Respected Chairperson, distinguished guests, colleagues, ladies and gentlemen, at the outset, I would like to thank Almighty Allah as well as the organizers of the Third World Traditional Medicine Forum, as well as Madam Amina Al-Haidan, Chairperson and the founder of Lotus Holistic Institute, Abu Dhabi, who has given me a chance to present among this august gathering and to deliver this talk, assuring safety of Arabic medicine, quality control, and standardization. Ma anzalallahu min da'in illa anzala lahu shifan. Almighty Allah has sent down, the messenger of Allah said, Allah subhanahu wa ta'ala has not sent down any disease except that he has also sent down its cure. Ever since the incorporation of traditional medicine in its program in the year 1976, the World Health Organization has gone a long way in creating a worldwide interest for the promotion and development of different systems of traditional medicine and their best utilization in the healthcare services. Let us have a glance at the origin and development of Arabic medicine. Well, the 9th and 10th centuries have been designated as the golden age of Arabic medicine. As the very name indicates, Unani system of medicine originated in Greece and is based on the teaching of Hippocrates, Dioscorides, Galen, Jaber, Razas, Ibn Sena, Ibn Baruni, Ibn Betar, Ibn Nafis, and so on, many other people. These philosophers and physicians have written a number of the books starting from 9th century to 13th century. And these books were very famous. I'm sure enough of you must have gone through Al-Qanun, authored by Ibn Sina, and Al-Hawi, written by Al-Razi. The Greek philosopher and physician Hippocrates, who freed the medicine from the realm of superstition and magic and gave it a status of science. He was the first person who established that disease is a natural process and the symptoms or the reactions of the body to the disease. He was the first physician and probably the only one on record since antiquity who introduced to take the medical histories. Hippocrates has written quite a number of the books. His chief contribution to the medical realm is the humoral theory. If Hippocrates systematized the medicine and gave it a status of science, Galen, the pioneer of experimental medicine and the author of 256 treatises, which were mostly medical in nature, in one of his famous books on anatomical procedures, which is a very standard medical text for some 1400 years, he advised to his readers, if you have to know a medicine, inspect it not once or twice, but frequently, and has given a very good example that as the twins looks alike to the strangers, but they are very well differentiated by the friends. If the Galen has stabilized its foundation on which the Arab physicians Ibn Sina, Ibn Razi, Ibn Betar, Ibn Nafis, Al-Dahrawi, Al-Baruni, and many others, they have constructed an imposing edifice. Well, plants have always been a common source of medicament, either in the form of traditional preparations or its pure active principles. In a survey done by World Health Organization, it has been estimated that 80% of more than 7,000 million, that is 7 billion inhabitants of the world, rely mainly on traditional medicine for their primary healthcare needs. and it can safely be presumed that a major part of traditional therapy involves the use of plant extracts or their active principles. Well, in developed countries too, plant-derived drugs are considered important. For example, in the United States of America, 25% of all the prescriptions dispensed from community pharmacies contain plant extracts. The World Health Organization noted that more than 126 plant-derived drugs are being used in the modern medicine, and the major pharmaceutical companies are now performing research on the plants for their potential medicinal values. The global industry analysts have noted that the medicinal plant sector has reached $125 billion by 2018, and they are expecting it $196 billion by 2025 and $5 trillion by 2050. But what is conveniently of the conservatively estimated 300,000 to 400,000 species of the higher plants that exist on Earth, almost 10 to 20%, that is 35,000 to 70,000, have been employed in traditional medicine. Well, the efficacy of these drugs is due to the presence of one or more chemical constituents. In the recent year, with ever-growing commercialization in the field of herbal Unani medicines, there has been an instant demand for the quality control of the Arabic medicine. For this, standardization is usually recommended as a solution to the problem. The studies on the identity, purity, quality of the genuine drugs will enhance information in checking the adulteration. A set of standards should no doubt be deterrent in checking the adulteration. Rather, it will be an aid to the drug law enforcement. But what is conveniently overlooked is that the standardization of the whole materia medica of Unani medicine is a difficult task. And therefore, before dealing with this topic, let us recapitulate what is meant by standardization. Standardization may be defined as elimination of unnecessary variety, or in other words, it implies the collection of the relevant data, putting it down in a logical sequence, eliminating the varieties and inconsistencies before formulating the standards. In order to appreciate the problems facing the Unani or Arabic medicines, we have to examine this situation in totality. Well, the term totality stands for examining the problem of standardization from the time of plantation of the drug, that is the cultivation, to giving it to the patients, either in the crude form or in form of the finished product. Above all, the identification of single Arabic medicine is a difficult task. Nowadays, adulteration is a common practice, and closely related medicinal and non-medicinal plants should be distinguished on the basis of their minute differences. Misidentification of two Chinese plants in a slimming preparation is believed to be responsible for serious toxicity, that is renal intestinal fibrosis, experienced by some 70 Belgian women. 30 of them needed dialysis or kidney transplantation. And this was due to between two plants, Stephania tetrenda and Astrolochia fanchai, and is thought to have been arisen due to the similarities between these two plants. Let us take the example of saffron, Crocus sativus. Saffron, that is the dried style and stigma of Crocus sativus, it contains three crystalline matter, alpha, beta, and gamma quercetin, having 0.7 to 0.3% of saffron, besides having refrigerant, tonic, diuretic, and stimulant properties. Saffron has anti-inflammatory, antiseptic, as well as aphrodisiac properties, for which it is being incorporated in many of the compound preparations. Since it's a costly ingredient, the adulteration is on its peak. To determine the authenticity between the adulterated and pure, the structure of the stigma, which is slightly expanded in the form of a narrow tubular structure, plays an important role. It's so costly, you can imagine that it is the most expensive agricultural product in the world, £5 to £10 per gram. And the substances used may be grouped in three categories, which are admixed. Substitution of other materials that have some external resemblance to saffron, exhausted saffron recovery by dyes, and the third is the substances added to saffron in order to increase the weight. There are quite a number of the adulterants which are being added to saffron. Be it pomegranate fiber, Carthamus tinctorius, safflower, Calendula officinalis, turmeric, curcuma longa. Curcuma longa is sometimes being sold in the market as Indian saffron, American saffron, and Mexican saffron. So there is a very simple identification test. If we test the material with sulfuric acid, it will produce a blue color, which will change to violet and then cherry red, and finally to brown. So this is a conformity test that this is a pure saffron. Similarly, if we take dimethylamine and sulfuric acid, first it will give a blue color, which will change to brown red color, and this will confirm that this is pure saffron. The FDA, Food and Drug Administration, they have given some criteria that the stigmas must be yellow, foreign organic compounds not more than 10%, volatiles and humidity must not more than 14%, and so on and so forth. Similarly, there are certain chemical tests provided by the United States Pharmacopoeia, and they test that the pure saffron should not include the yellow styles when processed between filtering. It should not leave an oily stain, and many others. You can go through it. The chemical composition of pure saffron is as follows. If we have an adulterated saffron sample as well as a pure saffron sample, you can see over here two adulterated samples and pure saffron, and the major protocol which differentiates between these two is the water and alcohol soluble matter, and you can see that it is above 50% in the case of pure saffron, whereas it is very low in the case of adulterated one. This is the proximate analysis of saffron. If we take the saffron and prepare the preparative chromatogram obtained from it, we get mainly 3 to 4 major constituents: crocin, picrocrocin, saffronol. And this is not only in one system. If we do the thin layer chromatography in UV light, in normal visible light, or spraying it with anisaldehyde, only these 3 to 4 compounds, they are visible and can be separated easily. These are, as I told, these are crocin, picrocrocin, saffronol. This the major compound, and its structure determined is the crocin. Well, pharmacognosy does not provide all the information, and standardization doesn't stop with the identification, but extends to quality and potency. Recourse to the physical and chemical method of standardization becomes necessary, rather a must. The chemical standardization is possible if we know the limits of variability of any characteristic constituents of the drug present in a measurable quantity. Compounds present in small amounts may not be useful for standardization purpose. The isolation of a single compound is itself a difficult task, and to press them into service for standardization purposes is even more challenging. Let us take another example of inter and intra species. Ami. It's Arabic called as khilla. Ami majas and Ami visnaga. That is khilla shaitani and khilla baldia. Take the fruits of Ami visnaga and Ami majas. Normally what happens that Ami majas is the cheaper one and is being added to the Ami visnaga. That there is a very good test that if we take the finely powdered visnaga fruit and add 1 to 2 drops of sodium hydroxide, it will give a rose red color. And this rose red color is not found in the case of Ami majas. So it's a very, very distinguished test. Besides that, if we cut the transfer section of the material, we found if Ami majas is being admixed with the Ami visnaga, it might be distinguished with 4 distinct secondary ridges, as well as the absence of lacunae in the vascular bundles. So this is also one of the very distinguished feature to be recognized. It can be checked by thin layer chromatography, by gas chromatography and mass spectrometry, where we found in AMI images a number of the compounds are there. The two major compounds found there are bergaptan or xanthotoxin. It is also called as 5-methoxysorelin or 8-methoxysorelin. But beside that, there are quite other compounds in small quantities like these. Whereas if we take the GCMS of AMI Visnaga, we found only two major compounds, that is chilene and visnagine. And these two compounds, if we do the high performance liquid chromatography and thin layer chromatography, it's a very clear cut picture that AMI Visnaga contains chilene and visnagine, and AMI images contains 5-methoxy and 8-methoxysorelin. So by the use of the chromatographic techniques, it's very easy to find out the number of the constituents present in various extracts of a drug. The RF value and the retention time determined under a specified condition is characteristic and can be used as an identity. The molecular weight and the fragmentation pattern will further facilitate the identity. Now, if an Arabic, let us say that the reproducible efficacy and reproducible safety will depend on the reproducible quality. And therefore, if an Arabic medicine wants to be regarded as a rational drug, it needs to be standardized and the quality must be determined. These are certain references for this particular reference. And the last, I would like to thanks, besides the organizers, I would like to thanks to Lotus Holistic Alternative Health Institute with a very special reference to Madam Amina Al-Haidan, the chairperson and founder, for granting her kind permission to present this paper. I'm really thankful to all of you for your patience through and through. Thank you very much.